She obtained her PhD from Bose Institute, Kolkata, where she explored the immunobiological mechanisms of T cell unresponsiveness during visceral leishmaniasis. Following her doctoral work, she pursued advanced research in USA at the Moffitt Center, Florida. She worked with Dr. Mark Alexandro on TGF-beta-induced cell cycle arrest via regulation of RB-MCM7 interaction. Later, as a research fellow at the Rocky Mountain Laboratories, NIH, under Dr. Karen Peterson, she investigated the innate immune regulation of La Crosse virus-induced encephalitis, a significant neurotropic viral infection. Dr. Mukherjee joined Presidency University in the year 2012 in the Department of Biochemistry, which later became part of the Department of Life Science. In 2019, she took charge as the coordinator of the School of Biotechnology at the Newtown campus of Presidency University, where she successfully launched the MSc Biotechnology program. Under her visionary leadership, the school evolved into the Institute of Health Sciences in 2022. Her pioneering research contributions have been published in prestigious international journals such as Molecular and Cellular Biology, Immunity, Journal of Immunology, Feb's Journal, Journal of Biological Chemistry. Her lab continues to receive competitive fundings from agencies like DBT, CSIR, WBT, STBT, reflecting the national and international recognition of her work. Today, we are indeed fortunate to have Dr. Mukherjee with us. Her journey reflects a rare blend of academic excellence, global research exposure, and visionary leadership. On behalf of everyone present here, I extend a warm welcome to Dr. Piyali Mukherjee and look forward to her insights and continued contribution to science and education. Ma'am, please, the stage is yours.
Is this on? Okay. So thank you very much for the very, very kind introduction. I'm very truly touched to be here, especially in front of people who I have known for a long time and who has been my source of inspiration. My journey was said from my PhD days, but I did my bachelor's in chemistry actually. I do hardcore biology research now. But Okay, thank you. But I switched to biochemistry, master's in biochemistry. The person for whom I am here is actually in front of me, and I am forever indebted to him for the constant inspiration. And you know, your teachers, you all know that they play a very important role in your. I can switch to the mic. It's okay. So that's fine. That's what I was trying to say, that our teachers play a very important role in our lives, and we become what we are because of them. So thank you, sir, for being here today for my talk and for inspiring us always. So today I'm going to talk about, as my colleagues said earlier about the path from mitochondria to nucleus in rotenone-induced neurotoxicity, and I will try to break up for all my younger audience all these terms. And because this is actually a part of my research, part of my journey, portion of my work for the past decade, there will be a lot of scientific jargons. So I would really urge you, if you have any confusion, I am also a teacher, so I really want you to understand at least to a certain extent of what we are trying to do. So, I have talked the good, the bad, and the ugly. So, sometimes in our system, how a good molecule can turn against us is something the story is about. So, if imagine a situation, I'm standing here alone and giving a talk. So that won't work, right? I mean, it won't mean anything to me, or there will be no one to hear it out. So this is exactly the situation within our body where everyone is talking to each other constantly. They're constantly interacting with each other, they're trying to understand each other and trying to support each other, and sometimes causing harm to each other. So that is what life is, that is what we see when we interact with each other, that is exactly what is happening within our cells. So I will go back and forth between different components of our cellular system, but I'll start with my favorite one. I love immunology, so I'll start with immunology. So you all know that whenever there is a foreign, our body detects a foreign molecule, they actually evoke an immune response. That means our body is fighting against the immune response. But how does our body actually recognize that it is foreign? So there are distinct patterns on the surface of different microbes, which are called pathogen-associated molecular patterns, or PAMs. And now that is very, usual stuff we all know since we were a child, that, if anything foreign is entering our body, there will be an immune response, and, our cells will, body will act accordingly. So that is fine. That is something we know. PAMP is something probably many of you don't know yet, but you will know eventually as you go through your immunology classes. Now the other stuff that I really want to introduce is the most fascinating part, which is very recently, like in the past decade, it has come up, and that is known as DAMP. If suddenly there is an injury to our cells, there are things that are coming out. Our own stuff that is coming out, but those things are actually foreign to the rest of the body because they have never seen it. They have been hidden inside a cell or in a compartment. And these are known as DAMPs. And they are damage-associated molecular patterns. And today, in most part, especially in the latter part of my talk, I'm going to talk about one such DAMP. So let me just give you an example of, a few of the DAMPs. You have all maybe have heard about reactive oxygen species. It may act as a DAMP if it is coming out within the cell. Think about mitochondria. You all know mitochondria. So mitochondria has a DNA. But it is our own, right? But if the mitochondria gets damaged and this DNA comes out into the cytosol, it is foreign to the cell and it evokes an immune response. So our own thing is, so mitochondrial DNA is sometimes, if it is coming out in the cytosol, it will act as a DAMP. So there is another molecule that lies within the nucleus that is called as HMGB1, or high mobility group box protein 1, which if it comes out, then it acts as a DAMP and stimulates an inflammatory response. Okay, so we will keep this thought up to this point. I'll come back to HMGB1 again. But the question is that why we got interested in what we are doing right now. Okay, so I did my postdoc in a neurotropic virus, and that is the time when I was introduced to neurons. Not only neurons, I have been doing host-pathogen interaction even during my PhD. I was really fascinated, but when I joined Presidency University, the infrastructure was not such that I could immediately start with a pathogenic molecule or anything like that. So it was a lot of settling down, a lot was building up the infrastructure. So, but I was very much interested. I became very interested in this fascinating organelle that is called the mitochondria. So I really wanted to understand. I had a molecule of interest. I wanted to understand that how the mitochondria behave in various neurodegenerative diseases. So I saw that there is a molecule that is known as rotenone, which is actually a mitochondrial complex 1 inhibitor. But it has been associated with sporadic cases of PD. Now what is sporadic? And PD is Parkinson's disease, and sporadic is the cause is not known. So 90% of the Parkinson's disease cases, the cause is not known. Only 10% it is familiar or genetic. So what does this mitochondrial complex 1 inhibitor has to do with the neurodegenerative diseases. So that is the questions that we started asking, and over the years we have been able to find out certain links like affecting the autophagy. I'll go through these, inflammation, loss of NAD plus, and then recently the cell cycle blockage. So this is an overview of what we have done, and I'll try to kind of cover a little bit about all these topics. So this is where the journey started. This is where it was feasible. So we started working on a neuronal cell line, very widely used neuronal cell line, that is SHSY5Y. And so that was because having a cell culture, and I'm sure many of my colleagues will understand and appreciate how difficult it is to establish a cell culture and do cell culture. In the meantime, I had very enthusiastic undergraduates at Presidency University. So they came and told me that, ma'am, we want to work with you. And I said, there is nothing, there is an empty lab. So, but, I said, I've always worked on mouse model, but it was not possible at that time to set up a mouse model. So I said, okay, can you just grow flies for me? And it was kind of mild weather, so we could grow them just in the labs. And I said, you know what? They used to buy this good and all these stuff and mix Rotenone in that fly media. And I was very fascinated to see right in front of my eyes, after a few days, the flies would unable to move. So that's how my Drosophila model of Parkinson's disease actually started. Then I, of course, subsequently bought the incubators and all. And very surprisingly, my first paper from Presidency was on Drosophila. So what we found was that when we actually exposed the aged flies with Rotenone, they actually are very much susceptible than the younger flies. So this was the study on age-associated neurodegeneration. But I have just kept the summary of this work just to show two things. First, that Rotenone actually induced neuroinflammation. Okay, neuro meaning in the neuronal cells. Inflammations are the production of certain pro-inflammatory cytokines, and also specifically the loss of a specific set of neurons. Those are called the dopaminergic neurons. Now that is also very fascinating in the case of if it is going to the brain, it should affect all the neurons that are there. However, they're affecting a specific set of neurons. Why? We still don't know. It is the dopaminergic neurons. So again, why, from what is the source of this new inflammatory molecules, and how is it causing the death of these dopaminergic neurons was the next questions that we asked. So we started actually with the cell death. We wanted to find out how Rotenone is causing death within these neuronal cells. So these are all in the neuronal cell line model. I have used several in the recent past, but I will just focus on one at this point. So what we used was, we did not go for a very high dose of Rotenone. We kind of chose a dose of Rotenone that induced like 40% of cell death. And you can see here that how the cells look like following Rotenone treatment. What I would like to point your attention is the condensed nuclei. You would think that these are apoptotic cells, however the story is very different. So what we confirmed in these cells was that yes, Rotenone indeed caused a loss in the mitochondrial complex 1 activity. It did not affect the complex 3 activity. ATP was going down, and as you can see that even the complex 1 protein levels, this is a cocktail antibody, so complex 1 protein level was also kind of going down. So this is like a validation, that okay, this Rotenone can be used in the neuronal cell line, and we can actually validate the model. So since I talked about, my title talks about mitochondria to nucleus, and I said that Rotenone is a mitochondrial complex 1 inhibitor, we wanted to look at the status of the mitochondria. So of course, these are all summary, there are a lot of other aspects to it, but I'm just trying to kind of compile the story together. So this is TMRM, which actually measures mitochondrial membrane potential. So so most of you know that mitochondria has an outer membrane and inner membrane, and the proton gradient that is actually formed during the transfer of electrons, it maintains this membrane potential. That means mitochondria does not get leaky and remains intact. So loss in mitochondrial membrane potential is one of the hallmarks of mitochondrial damage. So we found that yes, Rotenone actually, as early as 4 hours post-treatment, Rotenone induced a loss in the mitochondrial membrane potential. Of course, we did quantification for all this. And if you look at the mitochondria, so this is a staining with MitoTracker green, and you would see the long tubular structure. It kind of looks clumpy here a little bit, but these are very long tubular mitochondrial structure. But with Rotenone, they become very rounded up. And so all these are at a very early stage of rotenone treatment. Usually, people follow a end point, time point, like 24 hours, but we did a very, time point analysis of what is happening with rotenone treatment. On the other hand, you can see a increase in mitochondrial ROS. ROS is reactive oxygen species. We used a specific dye that kind of identifies the mitochondrial ROS, that is mitosox. So you can see an increase in mitosox. So naturally, if there is an increase in ROS, then one of the most important, most common thing that we do is we treat the cells with an antioxidant, that is N-acetylcysteine. However, to a surprise, although the ROS levels were going down, N-acetylcysteine or pre-treatment with NAC could not reverse rotenone-induced cell death. Again, we also used another very common approach, that we actually use the apoptotic pan-caspase inhibitor to see if apoptosis can be blocked. We did not find that as well. So it was not via caspases, it was not via ROS. Then what is happening? So this is where we started thinking that it is because, there is no one single point in a cell that can be targeted and things get back to normal. It does not happen because ROS is constantly generating, and one damaged mitochondria will lead to the leaking of this ROS to another mitochondria and damage it further, and this will be accumulation of damaged mitochondria. So if there is accumulation of damaged mitochondria, then what will happen? You need to remove this damaged mitochondria via the process of autophagy, and this is specifically known as mitophagy. Mitophagy meaning, or autophagy means eating up. So if there is some problem, they will clear the cells. Even in a house, if there are garbage piling up, think about the situation. If it continues to pile up, it will cause a loss in homeostasis. Similarly, in the cells, if there are certain damaged proteins or organelles which are accumulating, it will cause loss in homeostasis. So we wanted to see what is happening. What is the status of autophagy? We found that P62 was actually here the P62 is actually going up, and it was still accumulating, and you could see that when we transfected the cells with a GFP tagged LC3 is an autophagy marker, there were punctas forming, and P62 was localizing with this puncta. But usually when autophagy goes to completion, P62 will be utilized by the lysosomes and it will go down. It was not happening, but we really wanted to confirm. So what we did was we used a plasmid, which was a M-Cherry GFP probe. Now what happens is, in the acidic pH of the lysosomes, if autophagy is going to completion, in the acidic pH of the lysosomes, the GFP signal is lost, the green is gone. So what you will see? You will only see red cells. So if autophagy is completed, you will see red cells. However, we did not see, like in the DMSO treated cells, we could see this yellow punctas. Punctas are forming. That means autophagy is getting initiated, but it is not getting completed. So we did several assays. I'm not showing it here, to confirm this, and we used different autophagy inhibitors. What is happening? It was confirmed that autophagy is getting initiated during rotenone treatment, but not completed. Now many papers have shown that yes, rotenone induces autophagy, and that was big confusion for us. But when I looked at those figures, it did say that rotenone is inducing autophagy, but they never mentioned that it was not getting completed. So that brought us to the next most fascinating part of our journey, and we continue doing that, is that we were trying to understand that there is less ATP, because every function in our body needs energy. So there was less ATP. What else? So we were asking that whether these energy deficits are actually contributing to this defective autophagy machinery. So we found out several papers when we found that there is this very important metabolite, which is NAD+. It is the oxidized form of the NAD molecule. NAD you must be knowing, many of you. So what happens is that, NAD+, if you actually add NAD+, it will restore mitophagy, and in one of the disease like Werner syndrome, it could actually, kind of relieve the symptoms associated with this disease. So first thing what we did was we looked at the NAD+, NADH ratio in these cells following rotenone treatment. So this is a 24-hour endpoint, and we did find that there was a significant reduction in the NAD+ levels following rotenone treatment. But we didn't know what to do with this, because we had no experience, no prior experience of working with this NAD+ molecules, and we have no idea what is going on. So we actually searched the literature and found that actually depletion of NAD plus has been associated with several pathological conditions. It is not only neurodegenerative diseases, it was happening in cancers, in a lot of metabolic disorders, and if you actually follow the literature now, NAD plus has become one of the very important molecule that is associated with several diseases. So, but how does NAD plus will get depleted? How will it get depleted? And what is actually the role of NAD plus within our system? So there are very important, interesting. So this is the very important part of biology where we need to maintain homeostasis. Nothing can be used up too much or nothing can be produced too less, okay? So there are 3 NAD plus consuming enzymes which actually require NAD plus for its activity. So the first one is PARP, it is poly-ADP ribose polymerase, which is very much essential for repairing the DNA. If there is a DNA damage, then PARP actually goes to the DNA and repairs it by doing various things. But its substrate is NAD+. It absolutely requires NAD+. So think about a situation when there is continuous DNA damage, then what will happen? There will be constantly activation of PARP, which leads to hyperactivation of PARP, and there will be more and more consumption of NAD+, and then there will be loss of NAD+ in the system and energy deficit. Another NAD plus consuming enzymes is sirtuin. Sirtuin is a very important molecule. There are different isoforms of sirtuins in our cells, some in the cytoplasm, some in the nucleus, some in the mitochondria. They also requires NAD plus. During my postdoctoral years, I have worked on this protein. At that point, we had no idea that it is an NADase, but SARM1 is a pro-neurodegenerative molecule. If it is present within the neurons, it will actually kill them or cause axonal degeneration. Now, I think so in 2016 it was found that SARM is also not an NAD plus consuming enzymes, it breaks down NAD plus. So if SARM1 is activated in the cells, NAD plus will get decreased. So these are the 3 NAD plus consuming enzymes. So automatically, since we saw a significant reduction of NAD plus levels, automatically our question was, what is the status? Are these molecules playing any role? Today I'm not going to talk about the SARM story, I'm going to talk about PARP1 mostly, a little bit about the sirtuins. So what we did was we bought a few inhibitors. So we bought a PARP1 inhibitor, PJ34, sirtuin inhibitor, EX527, and we pre-treated the cells followed by rotenone treatment to see if something is happening, if these enzymes are playing a role. To our surprise, what we found, and till date, blindly, if I pre-incubate the cells for 1 hour with PJ34 and then treat them with rotenone, there is a fantastic reversal or prevention of cell death by rotenone. It is as robust. So this was a remarkable observation by us. We also did it in another cell line, unrelated cell line, HEC293, and it was happening, the same thing was happening there as well. So what you can see is here, the cell death assay, where you can see the MTT assay as well as we always try 2 different cell death assay in the lab. So that was suggested by one of the reviewers actually, and it works fantastic. As you can see that upon rotenone treatment, there's a very high, like around 40% cell death, and there is a significant reduction. We also adjusted the dose, significant reduction of cell death following PJ34 treatment, and this is also the phalloiding staining that kind of shows. Although the staining looks a little bit different, but when you look under the cells are very much viable. But is it true for all the other mitochondrial complex 1 inhibitors? There are also complex 3, complex 4, complex 5. So we actually treated the cells with all the mitochondrial complex inhibitors, pre-treated the cells with PJ34, and we didn't find any reversal of cell death induced by these mitochondrial complex inhibitors, indicating that it was very much specific to rotenone. Then what do we do with this? So then again, we wanted to see, as I said, it is very important, the timing of the, what is happening. So we already saw that there was an early induction of mitochondrial damage. So is this NAD plus depletion happening at that early stage? So what we found, yes, it starts as early as 2 hours post-rotenone treatment, and PJ34 actually could reverse that. To our surprise, what we found that when we used, or not to our surprise really, so when we're using a sirtuin inhibitor, as I said, we also purchased the EX527, sirtuin inhibitor could not actually reverse the rotenone-induced cell death. But we just wouldn't give up on sirtuin inhibitor just yet. There is a very interesting correlation. I will come back to later. But this is the scenario. This is where we stood at that point, okay? So how do we actually understand that, we see a depletion in NAD+, when we block PARP activity by PJ34, the cell death is reversed. But how do we actually show that PARP1 is indeed hyperactivated? So there is a very straightforward assay. We purchased a PAR antibody. PAR is the poly-ADP ribose that PARP actually puts on the molecules. So if PARP1 is hyperactivated, you will see this high molecular weight accumulation of PAR molecules. So first thing first, whether there is DNA damage. So first we actually did a Western blot with a gamma H2AX, and we found that following rotenone treatment, as you can see, there is increased H2AX levels. And then we actually did a time point assay with the PAR antibodies, when we found that there is parylation, actually increased parylation. It begins as early as 4 hours post-rotenone treatment. Now these kind of time-dependent assays have never been done. People have usually done an endpoint assay, so it was very important for us to really understand at what time point it is happening. I'm not telling the SARM story, but this time point is actually very important for these kind of cell death. So what you see is 4 hours, 6 hours, and 24 hours. We did this parylation assay, and indeed PARP1 was hyperactivated following rotenone treatment, and PJ34 actually, it completely removed that. So this was also very interesting to us, because if we treat the cells with PJ34 alone, the cells always look stressed. We acknowledge that because cells do not like PARP to be inhibited when there is no stress. So we have to acknowledge that. However, even in the presence of PJ34 or rotenone plus PJ34, this parylation is completely gone. But we wanted to cross-check whether PARP1 is indeed involved. So we knocked down PARP1, as you can see, there is a reduction, not completely gone, but reduction in rotenone-induced cell death in this SI-PARP1 cells. So that kind of showed that, okay, our hypothesis is going well, that PARP1 hyperactivation is probably causing the reduction in the loss in NAD+ levels, okay? So then what happens with the mitophagy? As I mentioned, that ROS is getting accumulated following rotenone treatment, because that's what we saw earlier. So we found one of my friend here from ISCS, Dr. Benu Brododash. He had this plasmid. So I asked for this plasmid from him, and it is a mito EGFP RFP. Same principle, but it is targeted to the mitochondria. So what you can see here, very beautiful staining. Yellow means that both red and green are merged. As you can see in the DMSO-treated cells, there are a lot of yellow, beautiful tubular mitochondria. In rotenone, the mitochondria gets shrunken or not very happy, but there are very few red dots. Red means that autophagy is happening. But when we pre-treat the cells with PJ34, at very distinct locations, we see there is increased mitophagy is happening, and this is an early time point. So PJ34 actually stimulated mitophagy, and probably it is not allowing them to accumulate these damaged mitochondria, because NAD+ is there to actually drive this process. So that was our hypothesis, and kind of this was the story that showed that PJ34, a PARP inhibitor, actually restores the mitochondrial homeostasis in rotenone-treated cells. How does this actually, what does this really mean in the physiological system? So we again went back to Drosophila model, and as I said, that NAD+ decline has been associated with various physiological conditions, more so in aging. And I said that the aging flies are very susceptible to rotenone-induced neurodegeneration. So in our brain, not in our brain, in the fly brains, there are 7 distinct clusters of dopaminergic neurons. As you can see, it is marked, and when you actually, it is not very clear here, but here, these are all dopaminergic clusters, and I have zoomed them, so you can see that the axons are intact in the DMSO-treated cells, but you can see that the axons are lost in the rotenone-treated cells in the fly brain. So this is actually staining with tyrosine hydroxylase, which is a marker for dopaminergic neurons. So you can stop me if you have any questions. I'm just going on. But what was very interesting to us, now this is not a very feasible, lot of questions have been asked in the past about this approach, and I still think it's not a very feasible approach to just inhibit PARP. What we did was we pre-incubated these flies. As I said, aging flies were more susceptible to rotenone treatment, so we pre-incubated these flies with a PARP inhibitor. Now, why we used a different PARP inhibitor? Because olaparib is actually a clinically approved drug for cancer. Okay, so it is kind of repurposing, but so we pre-incubated these flies with olaparib, and at 20 days, we treated them with rotenone. And actually these flies kind of die by 5 to 7 days, okay? So this is kind of 5-day treated flies. If you can see this beautiful, you know, all these dopaminergic clusters in the DMSO treated cells, and in rotenone these clusters are more or less lost. Actually it's gone. And this is just the olaparib. Just look at this. So even at 5 days post rotenone treatment, the flies which have been pre-incubated with olaparib, actually their neuronal structures were preserved. So this was a very interesting finding for us. It is there, it was done, this work was done by one of my very talented PhD students. She has she has completed her PhD 2 years back. And so we have this thought. So we had this thought that, okay, olaparib can actually reverse rotenone-induced neurotoxicity in a physiological model system. However, the problem is, as I said before, cells do not like to be treated with the PARP inhibitor. Think about a situation, because this is constantly happening. It is not happening that today only my DNA is getting damaged. It is a continuous process. So we need PARP. We cannot inhibit PARP throughout our lifetime and then, you know, think about that one day we might get exposed to something very toxic and then, you know, this inhibitor will play. No, that's not a feasible solution. But at least this showed that indeed PARP1 is playing a very important role, or PARP1 mediated NAD plus loss is playing a very important role in rotenone-induced neurotoxicity. So this is the part that I'm not talking, but this is where I want to bring, sorry. So this is where I want to bring back your attention that about the nucleus and mitochondria, because PARP1 is actually localized to the nucleus, but we started our story, rotenone actually kind of damages the mitochondria, right? So we see that there is a connection, but what is happening really, right? We still didn't know that how this is happening, because the idea is that, just give me 1 second. No, just the battery. Can you just put back the battery? Okay. So what is happening, hypothesis, how we concluded this work was that, you know, rotenone is causing mitochondrial damage. There is ROS production, of course, but more than that, there is accumulation of damaged mitochondria, which is perpetuating this ROS production. And this ROS is now kind of leaking out of the mitochondria, going to the nucleus, causing DNA damage. Because there is DNA damage, there is an automatic response of the cells to bring in PARP1 to repair that DNA damage, and this is leading to hyperactivated PARP1. And this hyperactivated PARP1 is actually leading to the loss of NAD+. Just for, because I want to talk about this a little bit. Now this SARM1, as I said, it is a pro-neurodegenerative molecule. It is an NADase, that means it breaks down the NAD+. However, it is also kind of inhibited, remains in an inhibited form in the presence of NAD+. So NAD+ is actually an allosteric inhibitor of SARM1. Now if PARP1 is consuming all the NAD+, then there will be NAD+ will get like kind of depleted from the cells, and this allosteric inhibition goes away and SARM1 is activated. And this is exactly what we have seen. I'm not talking about that because I really want to talk about the next part. So that's why, but this is a very beautiful, you know, correlation that we found, but the story was not completed, right? We wanted to complete at least to the best, yes sir. Yes, we couldn't. And we couldn't get, it should, but it didn't. And even we confirmed this in the flies also, the fly paper that I showed. So the same thing, we automatically thought that if there is ROS, then NAC would actually inhibit. So we fed the flies with NAC, and nothing was happening. So our worry was that probably we didn't know the dose of NAC that is required in the fly. So what we did in that paper was we actually treated the flies with arsenic trioxide. And you know, arsenic actually generates very heavy ROS. Even, and in the cells as well as the fly, NAC could beautifully, like just like a magic, it will reverse the ROS generated by arsenic. So that kind of gave us some confirmation that NAC dose is working both in the cells, and it has no role in this particular process. I'll come to your question actually. We found out, so I did literature search, actually recently found out that NAC can reduce the rotenone-induced ROS to a certain extent, but it will not prevent the rotenone-induced cell death. There was only one, No, and it is just one small paper hiding in a corner. I just fished it out for my paper, but that is the story. But it is, there are other inhibitors of ROS which are playing some role, and I'll come to this in the last part, because the NAC not doing anything was a big problem for us because no one would believe us. So we did multiple experiments. So we had it in the flies, we used the arsenic, and we had it in the cells. So that was convincing enough for us. For arsenic, it is like magic. If I treat the cells with arsenic trioxide, it will just be red with mitosocs everywhere. And if I pre-incubate it with NAC, it is just gone. It's like red versus black kind of thing. So this paper was published 2 years back. And I've been working with Rotenone for some time. I wanted to work on something else, but so we started different projects, and those are work going on in the lab. But while this work was going on, my another PhD student, he actually started some other aspects of Rotenone work. Because if you remember, if I go back to my fly model, there was this concept of inflammation. So we did not know that how inflammation is connected to cell death. Nobody still doesn't know. No one knows how inflammation is connected to cell death. Okay, there are a lot of markers, there are a lot of very intriguing kind of cell death coming up, and it is probably something that we are going to pursue in the future. But when we started, there was not, pyroptosis and all was just upcoming, So Saurabh actually started this work. Saurabh is my PhD student, final year PhD student. He has actually finished 5 years, and his joining coincided with COVID. So 2 years, no work actually could be done. So he started a little bit late in his PhD work, but he was really curious to study inflammation. He had this knack for immunology, so he wanted to study inflammation. So I said, okay, so now we know that PARP1 is involved, NAD+ is getting depleted. Is there any relationship between NAD+ and inflammation? So very interestingly, because we saw very early changes, like 4 hours, we could see a lot of changes. So we looked at all the pro-inflammatory cytokines. If you have studied innate immunity, you will know that IL-1 beta, IL-6, and TNF-alpha are the very key pro-inflammatory cytokines that is secreted from cells. But mind that these are neuronal cells. These are generally not immune cells. Now, another thing is that if there is a immune response, that NF-kappa B, which is usually in the cytoplasm, will go into the nucleus. Now, of course, we must, whenever I talk about NF-kappa B, we must pay a tribute to Professor David Baltimore, who passed away yesterday. So he's the pioneer in NF-kappa B studies. He did, whatever I'm saying is actually their findings. So NF-kappa B actually goes to the nucleus, and as you can see, that very nicely, most of the NF-kappa B were localized to the nucleus. So this kind of correlated with this pro-inflammatory cytokine profile in the neuronal cells. So, we were very excited because we had already had a target molecule. I told you that HMGB1 is known to be an inducer of inflammation. It actually comes out of the nucleus, then goes out of the cells, acts as a dam, because our body does not know HMGB1 because it is always hiding in the nucleus. Okay? So it will act as a dam. So we were very curious to understand that whether HMGB1 is playing an important role here. Now, why we got interested in HMGB1? Because in a cervical cancer cell line, HMGB1 was shown to interact with SARM1, which was our original molecule of interest. Granted, I didn't know anything about HMGB1 at that point, and even today, I don't know anything much about HMGB1. I really didn't like this protein very much. At one point, I was getting tired because I didn't understand it. But, life has different ideas for you. So we wanted to see whether HMGB1 is playing any role in Rotenone-induced sterile inflammation. Sterile inflammation means inflammation in the absence of any infection. And then subsequent cell death, because this would be the first time where Rotenone-induced inflammation would be directly correlated to, cell death. So we were very much kind of sure about our hypothesis that, something beautiful is going to happen. And yes, of course, we did see that, HMGB1 protein levels were going up. HMGB1 is getting released from the nucleus into the cytoplasm, but mind the time. I told you mitochondrial dysfunction, mitochondrial loss, all were, and PARP hyperactivation, all were happening very early, like starting from 2 hours, 4 hours. Here you see that HMGB starts getting out of the nucleus. These are just few fields, but to our head, we know that it really starts coming out from 6 hours. Mostly it accumulates at 12 and continues to accumulate 24 hours. Timings are not really matching. It is happening a little bit late. And that is how mostly the HMGB1 looks at 24 hours post-treatment. What you see is, again, look at the nucleus. The nucleus is very dense. And this is just the Western blot, kind of the nucleus and cytoplasm. Very tough Western blot to do. So fractionation also we did to see whether this time-dependent HMGB1 is actually correlating with the immunofluorescence data. So very good, right? So we hypothesized that HMGB1 is going to play a role in inflammation. Here we have HMGB1 coming out. We have inflammation. So good. Then let's see how HMGB1 is playing a role. So we went a bit further with the inflammatory molecules. We did all the 4, 6, 12, 24 hours time point with the real-time PCR. And as you can see, the entire inflammation went down. And this was repeated so many times by Saurabh because we wanted this to go up, up, up. And it never went up. And to our disappointment, we also saw that NF-kappa B was not getting localized to the nucleus at this late time point. So that was a time when we were really stuck in our journey. Already we had the COVID, and this was the hypothesis that we started off with. And this was a very disappointing time, very tough time for us because really lost. We didn't know what to do with this because, as I said, I didn't know this protein at all. I didn't like this protein, again, even more so. So, we just still decided to see that how HMGB1 is actually going out of the nucleus in the presence of rotenone. So we pursued our journey because we had to find out some way to find the answers, why inflammation is going down, but still HMGB1 is coming out of the nucleus. That has never been shown by anyone. So we first wanted to see what is regulating the translocation of HMGB1 upon rotenone treatment. Okay, so luck has it, or whatever, HMGB1 actually undergoes post-translational modification, and these post-translational modification actually drags it out of the nucleus. Now, what are these post-translational modification? The first one is oxidation, the second one is acetylation, and the third one is parylation. So now things started getting a little bit exciting, that parylation, again, PARP1 hyperactivation, parylation. Acetylation, regulated by sirtuins, because sirtuins are deacetylases. So we have certain kind of correlation with them already. So we first looked at what, again, we've started with the oxidation. And NAC, again, did not have any effect on HMGB1 subcellular localization. HMGB1 still going out of the nucleus in the presence of NAC. We did try different doses of NAC actually for these experiments. Coming back to PJ34, we could see that, of course, we know that PJ34 kind of prevents rotenone-induced cell death. It also prevents rotenone-induced HMGB1 translocation very beautifully. So these cells, all the HMGB1 were kind of localized within the nucleus. So the question is that yes, PJ34 definitely prevents cell death, so maybe HMGB1 is not coming out, okay. Whether this is actually functionally relevant, we had to find out. So what we did was, we did an IP, because again the parylation stuff comes and these IPs are not very easy to do. We did it several times actually, so because the PAR antibody is very expensive, so we try to preserve, but if you do an IP, your antibody is gone, really. So what we did is we actually pulled down with HMGB1. So if anything that is bound to HMGB1 will come out. So what we did was we IP with HMGB1 and did the Western with PAR antibody. And as you can see that this is the kind of molecular weight where HMGB1 is found. And you could see this intense, the staining pattern with the PAR antibody. So of course, this was not convincing enough. So we did the reverse IP, where we IP'd with the PAR antibody, and in IP, this small faint band is good enough. So we found this actually at the particular molecular weight, we could find the HMGB1, so which convinced us that, that HMGB1 is indeed undergoing parylation. So we also, I don't have that slide here, but we also showed that HMGB1 via IP as well as IF, that HMGB1 is actually kind of binding with the PARP1 too. Okay, these are all 24 hours time point. Now, what happens with the SIRT1? Because that is another NAD plus consuming enzyme. So what we found was that SIRT1 activity was going down in the presence of rotenone, and PJ34 restored it. Now, SIRT1, what happens is, now HMGB1 is undergoing acetylation. It undergoes acetylation and then it comes out of the nucleus. We already showed that it is undergoing pyrylation. What about acetylation? Because SIRT1 actually maintains HMGB1 in a deacetylated form. So if it is undergoing acetylation, that means it is away from SIRT1. So what we found, again we did an IP with HMGB1 and an immunoblot with pan-acetyl antibody, and as you can see again in this area where HMGB1 is actually should be there, so you can see increased acetylation over here. So what we did was we actually looked at the IF where we actually wanted to see if HMGB1 and SIRT1 are again actually coming out. We did the IP and all, but what we found was very interesting. Now SIRT1, as you could see, the activity is going down, the protein levels are also going down, but only in those cells. You can see SIRT1 in cells where HMGB1 is, when the cells are intact, SIRT1 is there. But when HMGB1 is coming out, SIRT1 levels are also going down. So we did this, distant mapping, my student did this in confocal several times. And cells which had HMGB1 in the extracellular, in the cytoplasm, also had SIRT1 also in the cytoplasm and very little levels. So this was a question that was asked by the reviewer, and I had to acknowledge that we really don't understand that how SIRT1 is actually getting regulated by rotenone. So that is our next question, but this is what we saw. Okay, so basically what happens, what is happening here is that in a normal condition, under homeostasis, HMGB1 actually helps, allows different molecules to access the chromatin, different transcription factors. So it is a non-histone protein. So it remains bound to the chromatin in the presence of SIRT1, which maintains it in a deacetylated form. Now, if SIRT1 activity goes down because of depletion of NAD+, then it will kind of come off. In the meantime, under DNA damage, there is more and more PARP at the DNA. So this PARP is also contributing to the pyrylation of HMGB1, and both these post-translational modification is causing HMGB1 to come out of the nucleus, right? So that was the hypothesis. So what we found was that it is kind of matching with our hypothesis that HMGB1 is post-translationally modified by pyrylation as well as acetylation, although we do not have a conclusive study on the SIRT1 story, which is something we would probably take up next, but this is what we found. Okay, so we had sort of answered about how HMGB1 is coming out, but what it is doing, how HMGB1 is related to cell death, or whether HMGB1 is related to cell death, because right now we know, by then we understood that HMGB1 has no role in rotenone-induced neuroinflammation, at least within the neuronal cell. It had no role. It may have in the immune cells, but not in the neuronal cells. So what is HMGB1 doing by coming out? What is happening? What is the consequence? So what we did was, as I said earlier, I don't know what is happening, just give me 1 second. What I told earlier, that please take a careful look at the nucleus. The nucleus looks very tight, It may appear that it is apoptotic, but it is not. So this is a feature of a mitotic nucleus, where the chromatin becomes very compact. So in one of the rack, one of my colleagues suggested that why don't you look at the mitosis? What is the status of mitosis? So we actually did a fax and we found that indeed there is a huge increase in the cells at the G2M boundary following rotenone treatment. So then if HMGB1 has any role in this rotenone-induced G2M arrest, that was one of the curiosity. No one has worked on it. There was no previous literature. It was just an assumption that we were moving forward with. So we looked around, and we looked around for inhibitors of HMGB1. Inhibitors will inhibit the activity. Then again, it will not make any sense. But we found one specific, very important inhibitor of HMGB1, that is glycyrrhizic acid. And to my surprise, recently I learned that glycyrrhizic acid is nothing but Joshti Madhu, So, but I didn't know all these things at that time. So glycyrrhizic acid, what it does, it keeps the HMGB1 within the nucleus. It will not allow it to come out of the nucleus. And what we found very beautifully, now what you see, this red, it is actually stained with phosphohistochemistry histone, and this is his phosphorylation at serine 10. So it is H3S10 antibody. This circle shows that the cells are arrested at a particular prophase of the mitotic cycle, and this is HMGB1 that has come out. In the presence of glycerizic acid, HMGB1 remains bound to the nucleus, and there is no G2M arrest. So this was a eureka moment for us that, okay, we are kind of finding a non-canonical role of HMGB1, but I was very, very nervous because it's a big claim, and it is, HMGB1 is a very famous molecule. Although I knew very little about it, I learned that it is a very famous molecule. To make such a claim, I need to have a lot of backups. I need to have a lot of studies. So what we did is we first looked at some of the molecules that is accumulated at the G2M boundary, okay? So cyclin B, securine are these proteins which, cyclin B, CDK and all those cell cycle proteins, if it is kind of accumulating, then the cells are arrested at G2M, which we actually saw. This is a very robust Western. We have done several times with different treatments. And we could see that it is not completely gone though, okay? It never happens. It cannot happen like magic that, you add something and it's gone. But it is getting significantly reduced in the presence of glycerizic acid. I must tell you that the reversal with PJ34 is more robust always than with glycerizic acid. So PARP and the NAD plus depletion is the key player, but this is also a very important player. So this is what we had. So basically, what we realized is that when HMGB1 is in the nucleus, under stress, it is undergoing parylation and acetylation, going to the cytosol, and this event is causing the G2M arrest. If I hold HMGB1 at the nucleus, does not allow it to leave, then the G2M arrest is not happening. Okay? So, but how this is happening is something, we kept on asking these questions, but if it is true, because as I said, it is a big claim. So what we did was, we looked at the HMGB1 protein and found that there are two NLS. Okay, and these NLS has lysine residues, which actually gets acetylated. So what if we actually make some deacetylate mutants? Then they will not actually leave the nucleus. So we did two different mutants where we changed the lysine to alanine or arginine. And as you can see, this is the full length HMGB1 where it is actually in the presence of rotenone, it is going out, and there is arrest at the G2M. And this is the two mutants of HMGB1, actually three, I'm coming to the third one, where they are unable to exit the nucleus. As you can see, very dark green staining. That means they are kind of retained in the nucleus, and even in the presence of rotenone, these are not coming out, and there is no G2M arrest. So this made us really, really confident and very, very happy, of course, that okay, so we did find that HMGB1 has to play some role in the rotenone-induced G2M arrest. We also used another mutants where I have the NES, the nuclear export sequence was also mutated. This was suggested by one of my colleagues. And so we did that too, but that is a mixed response. For some reason, we see both. Like here, where in this NES mutant, where you can see that HMGB1 is enriched at the nucleus, it is not coming out and there is no G2M arrest, but there are certain populations where these are happening. So it could be that there are other putative NESs within the HMGB1, which we probably don't know about at this point. So we didn't leave it at that. We wanted to really go a little bit forward and understand what is happening, how HMGB1 is getting regulated, or, what is the crosstalk between mitochondria, cytosol, and the nucleus. So the last thing that we did, actually, it made a lot of difference, actually. So cell cycle. Cell cycle means there is a lot of changes that is happening within the cytoskeletal structure of the cells. Rotenone is known to bind tubulin. So out of curiosity, in a very isolated experiment, we have once done this tubulin acetylation staining. So we just wanted to show that, yes, the cells are getting destabilized and tubulin is getting depolymerized. That was our intention of doing this tubulin acetylation staining as a backup to our cell cycle arrest. What happened was, we constantly found, as you can see in a time-dependent manner, as early as 4 hours, the tubulin is getting hyperacetylated. Now, as I said, predominantly rotenone, HMGB1 starts coming out at 6 hours. But tubulin is starting, so this is again a very robust assay for us. We have done it several times. So tubulin is getting hyperacetylated. I am not an expert. I didn't know what this means. So one of my colleagues, Shubhro, he actually works on these stuff. So he came forward. What he did is said, Piya Didi, why don't you actually block this acetylation of tubulin and see what happens in the cells? So he gave me this siRNA of TAT1. Now TAT1 is tubulin acetyl transferases. So what we did was, we first tried to knock down TAT1. So this was a very established primer which had been previously used in a paper. So we used it, and because the reviewers just wanted to see it, we had to do the real time this time to show that yes, when I'm treating the cells with SI-TAT1, the TAT1 levels are going down. Okay, so we are seeing tubulin acetylation, and now we are transfecting the cells with a molecule that will prevent this acetylation. So what is the consequence? So first, of course, we confirm that in the SI-TAT1 transfected cells, the cells again look dark. There is only blue staining. There is no red staining of acetylated tubulin. But what we found very interesting, I actually didn't want to believe it at first because I couldn't explain it. Sometimes we do that. We are sometimes when you can't explain something, we kind of want to skip it. But my student, Shourabh, was adamant. He was like, no ma'am, there is something going on there. So what we found was, in these SI-TAT cells, HMGB1 was not going out of the nucleus, and it was retained within the nucleus. Okay, and there was no H3S10 positive cells. No meaning, I wouldn't say no, but it was like majority. Majority of the cells, as you can see, majority of the cells were H3S10 negative. So this was, again, we couldn't explain what is happening. How these things are getting connected was something we didn't know. We were scratching our heads because we see everything in a very, isolated manner. Okay, tubulin is getting hyperacetylated. There is mitochondrial ROS. There is PARP activation. HMGB1 is coming out. There is cell cycle arrest. Too many scattered things. We couldn't really connect. So then actually we went back to ROS. And sir, this is where the next part is that NAC is unable to reverse ROS production. What we found was that in these SI-TAT cells, there was reduced mitochondrial ROS both at 4 as well as 12 hours. But we couldn't really, but then what is happening? If ROS is getting reduced and SI-TAT cells are actually being able to recover from rotenone-induced stress, then the NAC result was still not, we couldn't really wrap our heads around the NAC result, the N-acetylcysteine. So we used, another colleague suggested that why don't you use this ROS inhibitor, which is DPI. It is actually an NADPH oxidase inhibitor. And DPI is very tricky. The dose of DPI 5 micromolar, it killed all our cells. So we had to be very careful with the choice of the dose of DPI. At 2.5 micromolar, DPI actually reduced not only rotenone-induced cell death, but both, I have not shown the pictures, both G2M arrest cells arrested at G2M as well as the HMGB1 translocated cells. So you can see that it is not NAC, but it is another ROS inhibitor which could actually reverse this to a considerable extent, and this was also prevented in the absence of tubulin acetylation or hyperacetylation, whatever we call it. So there was this connection. Okay, so we again continued with this to see if this is indeed true. So we wanted to see now the status of tubulin acetylation with the glycerizic acid, because glycerizic acid does not allow HMG1 to come out. But as we see that there is reduction in the G2M cells, in the H3S10 positive cells, which means that there is reduced G2M cells in the GA treated cells, like glycerizic acid treated cells, but there is no change in the tubulin hyperacetylation, which kind of told us that tubulin hyperacetylation is preceding HMGB1 nuclear exit. So even in the presence of rotenone, even if HMGB1 is bound to the nucleus, tubulin might get acetylated more, okay? But it just cannot execute because HMGB1 is present within the nucleus, which really puts HMGB1 in a very important place in rotenone-induced neurotoxicity. So again, then if we talk about DNA damage, because DNA damage will bring in more PARP1, which will cause reduction in NAD+ level or pyrylation of HMGB1, then what is the status of DNA damage? So these are all the, the cells at different stages of mitosis, and you can see that this is gamma phospho H2AX staining, which actually goes and binds to the double-stranded DNA breaks, and you can see that even in the presence of rotenone, there is an increase in the H2AX positive cells, which I showed showed in the Western in an earlier slide. But in the SI-TAT cells, there is a significant reduction in these gamma H2X puncta. Now coming to GA also, you could see that there is a reduction. I will not say it is completely gone because DNA damage is constantly happening, but there is a significant reduction in the gamma H2X positive cells, which shows that, you know, when HMGB1 is enriched at the nucleus, rotenone cannot really induce such, or the DNA is getting repaired. I cannot say that conclusively that rotenone is not inducing DNA damage. Probably the DNA is getting repaired efficiently. So we wanted to see now nocodazole. So all of these experiments, I did not show those data, all of these experiments were done in parallel with another G2M blocker, that is nocodazole. Nocodazole, it's a very beautiful molecule that will arrest the cells specifically at G2M. Because rotenone was also arresting the cells at G2M, we use nocodazole for all our experiments. What we wanted to see that whether the cells are able to recover from rotenone, because nocodazole in cell cycle, if you do research in cell cycle, you will always use different inhibitors, you know, to bring all the cells at one stage of the cell cycle and then release. So with nocodazole also, if you remove nocodazole, the cells get released into mitosis, okay? So, you know, we also wanted to see if rotenone is also doing the same thing. So we did what we did, 24 hours we treated the cells either with nocodazole or rotenone and then released the cells for 12 hours. That means we removed the media containing nocodazole, washed it, and then added fresh media which did not have any of these. As you can see, this is the nocodazole, very comparable levels of G2M arrest. When we release, you can see the cells are, you can even look at the cells, the nucleus also, very beautiful. They have come back, they have recovered. But this could not recover. So rotenone treated cells could not be recovered from this stress, and so this is a phenomena which is known as mitotic catastrophe. So when cells are actually arrested at mitosis and cannot proceed further. So what we found also after this post-release, there was, when you can see that the nocodazole-induced DNA damage is comparable in both rotenone as well as nocodazole-treated cells, it is getting kind of recovered. However, in rotenone-treated cells, these are still going on. What does it mean? So this is the last slide, at least of the story. What happens is, we wanted to see if HMGB1 is playing a role in the DNA damage repair process. That means if PARP1 via increased pyrylation is knocking off HMGB1 from the chromatin, then probably DNA damage repair is unable to occur. Now this arrest at G2M is a natural response of our cells. That means cells always pause at different stages of the cell cycle to assess, okay? Assess whether there are any damage. Once they have assessed the damage, they will bring in molecules to repair the damage. If the damage, because if the cells proceed with this damage, then you know that tumorigenesis is one of the consequences of that. So this G2M arrest in the presence of rotenone is a natural response of the cells. So it is nothing bad, it was the good. But that turned into bad and later ugly by continuing with the cell death process. And HMGB1 is probably an important player because what we found, we actually did reanalysis of the ChIP-seq assays. ChIP-seq meaning, ChIP means chromatin immunoprecipitation, where people find out which is binding to which region of the, whether they are binding to the DNA promoter region of different genes. And in those reanalysis from previous papers, we found that HMGB1 actually binds to various DNA damage repair genes. So we found those names, we ordered those primers, and we looked at their expression at different time points, 4 and 24 hours. What you can see that at least for some genes, they were trying to get, respond to this DNA damage, but however at 24 hours, the expression of all these genes were actually not happening, which should have happened because there was an ongoing constant DNA damage. So this was not happening. So all of these kind of put together that the presence of HMGB1 in the nucleus is very, very important to kind of bring back the cells to homeostasis in the presence of this ongoing DNA damage. And if, because the cells cannot do this because HMGB1 is coming out, it is leading to this cell cycle arrest and subsequent neurodegeneration because of the mitotic catastrophe. So basically, this is the summary of this entire story. What we believe is that rotenone is causing this binding to tubulin, resulting in tubulin hyperacetylation. There are papers which shows that ROS induces tubulin hyperacetylation, but it could be a cyclical event. And this ROS is now going into the nucleus, where it is causing DNA damage, leading to all the events that I mentioned, and then it leads to the coming out of the HMGB1, that is subsequently leading to the G2M arrest. So this is where the story stands now, but the question that has been bothering us is that it is a cycling cells and neurons are terminally differentiated cells. So at this point, what Saurabh is doing is actually, we have been able to kind of differentiate these neurons into post-mitotic cells, and we are looking at how this, because, I'll come to that. The other thing that I did not mention is we have also done all these experiments. We had to. The reviewers made us repeat all these experiments in another neuronal cell lines. We used PC12 cells, and just out of our curiosity, we also repeated this experiment in microglial cells. But in microglial cells, in the presence of rotenone, there is no release of HMGB1 from the nucleus. So that was a striking difference from us. And I'm happy to state that this paper was accepted in JBC like 2 days back. Final acceptance came. And so what is the implication of this study? As I mentioned, the neurons are terminally differentiated, but many papers, especially in Alzheimer's disease, people are showing that despite being terminally differentiated or post-mitotic nature, neurons can re-enter the cell cycle. Now, if they re-enter the cell cycle and these damaged state continues, then they will not be able to recover, and these will actually lead to cell death. So this is the part that we are kind of trying to just wrap up and understand, that how this entire process that I just mentioned is actually leading, whether it leads to the death or the loss of recovery from the stress in the post-mitotic neuron. So that is the next question, because tubulin depolymerization is also becoming a key player in neuronal degeneration. So probably these are the connecting dots that is happening. We still don't know, long way to go, but I just want to finish that part of the story and then move on to other stories in the future maybe. But this is where we stand right now. So of course, no work is possible until and unless you have the support of your students, of your institution, and your collaborators, your friends, your family, and several people who provide you inspiration. So this major work was done by Saurabh, who has really worked hard. Saurabh, I just cannot say how hard he worked. And these are the other members. Shemanti also contributed a lot, helped him a lot in finishing this work. These are the past members. Shubhro and Shom Shubhro actually contributed intellectually a lot on these papers, because many of the stuff I was not very well acquainted with. I also learned a lot from this work. So this is some of my past members. We need to click a new picture. These are my students. This is the building that you see right beside you. So we are there. And so my lab has been funded, thankfully, by several funding agencies. I currently have a DBT, CSIR, and WBDS-TBT grant with different other exciting work that my students are doing. So thank you for your kind attention.
Thank you, Pyalidi, for such a fascinating presentation, and also congratulations to you for the JBC paper. So any questions from anybody? So if you can have any questions, you can ask students. Any questions from the faculties or students? Yeah, please.
Yeah, ma'am, very impressive talk, and actually very informative also. Just a simple question, ma'am. Ma'am, why retinol and why not LPS or something? Because I have used this SHS5 just for this repurposing of glioblastoma. Like, I wanted to use that. And I have used, basically that was all in vitro only. We did not go in this physiological model and all. So initially we started with H2O2O. And then we went to this LPS, and it showed all the markers. It was all in an initial level only. So what, can you just throw some light on that? Why detonone and why LPS?
That's a different question.
Right.
So LPS is a part of the gram-negative bacteria. So like, we can get infected with different bacteria. Bacteria usually don't, except for certain exceptional cases, they don't usually cross the blood-brain barrier. So, so studying with LPS in the brain was not that exciting to me at that point. However, rotenone crosses the blood-brain barrier. It is implicated in Parkinson's disease already. And rotenone is just one case of pesticide. Think about the environment. Think about what is going on. We don't know what we are consuming, okay? So one of the other work is there on a rotenone fly model where I like at this point, at this stage of my life, whatever we are doing is going to get affected in the later age. That is what the realization is. So the question was that how the environment is actually leading to this neurodegenerative process because we are constantly exposed. So rotenone is just an example. If given a chance, I would like to work with many other pesticides, like organophosphates and all. But so much of tedious work with one, it might take a long, but that is the most next relevant question. And that was the only thing that because, and the other thing was that rotenone is a mitochondrial complex 1 inhibitor. So I really wanted to see how mitochondria is playing a role in these neurodegenerative processes. Now with LPS, it's a very different questions. I do work with a neurotropic virus, thanks to sir also, that is Chandipura virus, but that is a neurotropic virus that actually crosses the blood-brain barrier. And it infects the neurons, specifically. So that's why I have been working on rotenone. It is not for any specific reason. And the other thing is, if I would want to work on LPS, I would use a microglial cells because LPS has a more robust, induction of inflammation and all that in a microglial cell population than in the SHSY. I have never used LPS in SHSY.
I have used that.
Okay.
Also in HEC also, HEC, this fibroid cell and SHSY.
Okay.
And it showed all these markers like IL-6, everything was like...
Because that is inflammation. LPS will do that. LPS what it does, it actually goes via the TLR4, causes NF-kappa B to go to the nucleus and then turn on the pro-inflammatory cytokines. So LPS is a very known, but I am sure the robustness will vary between any immune cells and HEC and all the neuronal cells. But yes, but my question was different. My question was not infection-induced, all these changes, but environmental toxin-induced neurodegeneration. So that's why. Okay.
Thank you, Piyadidi. It was a beautiful talk. I have just one question that we all know that ROS, reactive oxygen species, can cause membrane damage. That is also reflected in the mitochondrial membrane potential depletion. Now, is it possible that ROS can also damage the nucleus membrane also?
Membrane, yeah.
And whenever it will get damaged, then the HSBC1 translocation will be possible, as well as we all know that damage of nuclear membrane can also cause that G2M cell arrest. So is there any relation between these two?
That's a fantastic question actually. That's what we thought. That's exactly what we thought, that it is because of all these damages, this is just coming out. It is when we did the experiment with the mutants as well as glyceric acid, we got... Yes, so that was the time, because that was the question, not only me, everyone was asking. I did a faculty seminar, we have a faculty seminar series, and everyone jumped on me with these questions only, that how do you know that it is not a consequence of loss of nuclear membrane integrity, that HMG1 is just leaking out. So when we actually blocked its leaking out, even in the presence of rotenone, see, tubulin is getting acetylated. Okay, so it's that those prior events are happening. When we blocked it and the G2M arrest was blocked, that gave us confidence that it is not just a mere consequence, it is something very mechanistic, that it is playing a very active role. The cheap qPCR that we plan to do, actually it's in the process, will actually confirm that it is, if we can confirm that it is binding to those, DNA repair genes, I think so that will give us a more robust understanding. The reviewers did not ask, we just continued all the other cell line study, but that is something Saurabh is going to finish up before his thesis actually. So, but that was what we were thinking. That's why we were very sad. That's why I felt that my science is going nowhere. That was a stage of my career where I was really frustrated, because I'm thinking that I've been spending all these 2 years on HMGB1 and, it's probably just, cells are getting damaged and it's coming out. But those mutants and those glyceric acid, all these assays, they actually made me confident that no, it's not just, But, that's very relevant, and it may happen. It is probably happening. There is some leaching out probably that is happening because of the damage. I have no idea how ROS will damage the nuclear membrane. I have no much idea about that, but you know.
It can damage by consecutive dye information in the
Acha.
lipid.
Acha, lipid, okay. Lipid peroxidation or something like that, okay. But that is very interesting question. That's something we were very frustrated with too.
Anyone else? Students? Any question? So Pierre, I was just thinking one thing, if in your initial slides, once I saw when you are showing the cell model, the PJ34 that you are using along in the cell culture itself together with the rotenone. And in the fly model, it is pre-exposed with the inhibitor.
No, even in cell culture also we don't use it together. I mean, we have done it
okay.
together. The reversal is not as much if we add, so we have done 1 hour pre-treatment, we have done co-treatment, and we have done 1 hour post-treatment. Post-treatment reversal is not that much. Co-treatment also, it is there, but it is less than the pre-treatment.
Oh, so the cell culture itself also we have 3 different.
No, but what you saw in the Drosophila model is very remarkable. I did
Okay.
not just because it's
Okay,
too much,
okay.
too long, you know. So we have done pre-incubation. We have also done co-treatment.
Acha.
Co-treatment does not do anything. What I keep on telling people now is that, you know, right now we should check our diets, check our lifestyle, because pre-treatment we cannot do. I cannot pop in olaparib pills, I will die. Okay, I cannot block PARP. But what we do now is actually getting reflected in our age, because we found that if you give medicine when the symptoms have started, it will not work. You have to have your body kind of
Already, okay.
kind of, yeah. So that is no. So pre-treatment, we also have the co-treatment of olaparib. I just did not show it because
Okay.
of the too many things, you know.
Okay. Okay, thank you. So if nobody else has any questions, so maybe I would request, ma'am, you can take seat, and I will request our Assistant Director, Dr. Rajat Pal, to please come up and give the concluding remarks as well as the vote of thanks.
Yes, and so good afternoon, everyone. Yeah, a very amazing talk. I have seen, probably Saurabh has given this lecture in SBC. Yes, and there I thought that it's so nice work Pialidi is doing just beside our building. So what can I say? It's really very, very good work and very good lecture, as she is very popular for giving lectures. So thanks, Pialidi, for coming here and giving us this insightful talk. To my students, thanks for the gracious presence of Professor Chattopadhyay, our Pro-Chancellor. And I'm very much thankful to Anindita. Yeah, she is actually the man or woman for arranging this. And it's fine. Probably we are going to meet in our next session of this lecture series, distinguished lecture series. So for then, bye for today, and we shall again meet in the next lecture series. So bye for now.